This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
Paul Verschure here for the Convergent Science Network podcast, together with Tony Prescott. And today we're talking to Barbara Finlay, speaker at our BCPD Summer School 2015. So Barbara, you've been very focused on understanding brain evolution, and also you emphasize issues of, let's say, the segmentation of brains, the commonalities between different brains. But what do you see as sort of the underlying principles of brain evolution?
A very large question to start with. Okay, so I think the underlying question that I've come down on is the one that that emerges from a lot of research in evolution and development at evo-devo field, which is how do you design something that is adaptable, that is responds to change, and robust, does not respond to change simultaneously. So that's how I've started characterizing how to best understand how nervous systems evolve. That is distinguished in my mind from the kind of random walk model of evolution that people have had to a large extent in behavioral biology so far, which is to imagine a brain and an organism as a series of ad hoc evolved patches as opposed to a set of theme and variations on this adaptation and robustness.
Is this what you mean when you talk about the filter idea?
Yeah. So if you think about evolution of the brain only in terms of adaptation and maximizing, say, how well you can detect a particular set of flowers or evade a certain predator or something like that, you come up with particular kind of nervous system that you imagine as being optimized for all these very specific kinds of behavioral problems. But if you imagine that the same nervous system has to also survive horrible catastrophes and shifts of niche and all those sorts of things as also part of its repertoire, you come up with a different kind of nervous system that you need to evolve rather than this sequentially adding of components. You come up with something that has to have a certain set of sort of core survival recognition learning functions as part of its repertoire, not only a set of maximum adaptation.
So is it then you're thinking less about evolution on the species level and more about phylogenetic change?
It pushes you in that direction. I mean, all the causal structure is going. The causes eventually we're going to come down to the survival of this versus that individual. But I think the idea is to think of cumulatively over individuals, both the sort of adaptive maximization kind of things. Can I recognize that particular kind of spot? Or, can I find this kind of food the best versus can I find any food? Can I get out of this hole? That kind of. So we've got two kinds of individual histories to amass, not just the adaptation one, but also the, dealing with any eventuality one as well. And the ones that remain alive have to do both.
So that would be the fitness aspect of it, like how appropriate is the behavior coming out of that system in the end. But then, so what you emphasize actually also during your talk, well, let's say 4 key perspectives, let's say that you follow, right? One is this whole idea: how do you allocate neural structure or neural modules? However, we can talk about how to define that. But how do you allocate those strategically in the face of these challenges for survival, right? And then the second one, of course, how do you then exactly control the parameters of your neurogenesis? The fourth one was how do you get coordination to actually build a multi-component system? And lastly, this whole role of embodiment and motivation as additional constraints on that process. So these are four big categories, if you want, of questions when we look at this whole issue of the development of a species-specific brain. So, but now if you would have to rank order those as into which of these four categories would be understand best, and which of these four is actually of the greatest importance for understanding, how would you rank order the four?
Let's see. I hope we get to edit some of this out here, but
no problem.
Yes. Those four things I talked about were more how the sort of empirical gathering of data that I did, separated itself into categories. The real categories, I think, span those. So there's the one category which Jerison called, I think, originally proper mass or something, which is allocation of neural resources to what the animal's actually going to encounter. And that is both point number 1 and point number 4. Okay. So we find what looked like something of easily modifiable or presets or something like that in an evolutionary sense of, am I going to be the kind of animal that maximizes chemosensation or visual auditory or something like that? And it seems to be that over and over again, most likely independently from the very first bony fish and sharks, amphibians, reptiles, mammals, that animals keep diverging on those same differences again and again as a gross allocation of neural mass to one or the other in terms of processing resources. Okay. But then the last bit of the talk, which is that I'd say probably the most of that, given that bias in where you're going to get your sensory information is going to be coming from how the environment and motivation instructs the animal. So in terms of nervous system content, that's the big thing. Now the other two are, I think, both about processing, and those are orthogonal. So my covert theory about what makes a vertebrate and why vertebrates took off at the 450,500,000 years it did is they had in place sort of the four big learning engines that would be useful. So this is which is things like cortex and hippocampus are both sort of association information extractors, and then we have reinforcement learning is embodied in basal ganglia, and the last sort is the cerebellar kind of learning, which is comparison and subtraction and optimization on that dimension. You find all those in the very first vertebrates all together.
And that's a bit of a surprise, actually, even to many professional neuroscientists who will say, oh, cortex, that's just in mammals.
Well, I don't mean the cortex per se. I mean the thing that
the thing that cortex is doing is in all these other species. But your talk emphasized the conservation of these structures
exactly
over time, but also the adaptability, and the adaptability was particularly around the relative sizes of parts of the brain. But even there, the single greatest predictor of how big any bit of the brain is going to be is how big the overall brain is. That's right.
Yeah. So each part of the brain has its own rate of change with respect to total brain size, and apparently no vertebrate has ever decided that the way to enlarge your brain is to, double down on the number of motor neurons you have and nothing else. So
Right.
what animals do, if their energetic situation or whatever allows them to support more brain, where that extra brain is going to go is in these sort of laterally placed association areas that look across sensory and motor systems.
And so there's an interesting question here around, whether no vertebrates explored outside that space because it's not possible to go outside that space, or it wouldn't be evolutionarily advantageous, or is it just that there's certain aspects of this brain architecture which are locked in place, which cannot now be changed easily?
Yeah, that's a very interesting question. Paul Katz, who is at Georgia State University and past president of the Society for Neuroethology, had some data recently that just blew me away, which was sort of going to this particular question. So he studies marine mollusks, and of which I'm going to do violence to the numbers on these, but let's say there are 60,000 of them, give or take an order of magnitude. Okay. And of those marine mollusks, only a very small fraction, some number, say a couple order of magnitude down, say 40 to 100, actually detached and have become mobile. And given the kind of work I've been doing with vertebrates, I fully expected to see that we would see sort of bilateral symmetry and swimming along by alternating contractions, and the kind of the vertebrate optimal I had come to expect from seeing all those different kinds of things. What you do see couldn't be more different. So we have all these animals that have been attempting to swim for comparable periods of time, and some of them are doing handsprings. Some of them are just looking like they're having some kind of seizure. Some of them are doing and their job is, it's just like the evolutionary learning algorithms where they make animals in the computer reproduce if they're just succeed in getting over some finish line. These are the same kind of things, and you see this same kind of wild differences in kinds of movement. And then I realized on seeing that stuff that truly not everything is evolvable.
Right.
that these are animals that have become mobile and have, but
it's not a good starting point.
Yeah, they've remained there as sort of singular and kind of comical examples, a lot of them, and so there is some data that would bear on this kind of question about what kinds of things permit changing. I guess if you read the classical account of, and by that I mean there's a book about Romer, which everyone reads when they study vertebrates, and what they attribute that the sudden change in the number of extant species from 30 species to 40,000 species of lamprey-like animals to bony fish, they attribute that to the jaw.
Yeah,
that you can now exploit so many different kinds of prey and eat so many different things. Well, jaw's all good, probably true, but there's a brain there too that suddenly appears.
Yeah,
and
but the brain isn't so different maybe between ancestral jawless fish and the successful jawless fish.
There's not so much forebrain. That the whole parasympathetic sympathetic division of the nervous system doesn't really come in till you get the jawed fishes. So there's been a lot of,
but we don't really have any extant jawless fish that are of any sophistication. We have scavengers and bottom feeders.
True, but it certainly could be the case that there could be combinations that we haven't seen, but what we have there, the ones that did succeed, that have populated all these niches, do have extra brain features as well, as just the jaws. And so I'm not saying that I know that to be the case, but there's sort of 2 points I'm trying to make here. One is that that once you see Paul Katz's catalog of marine mollusks, you can see that without any particular proof at this point, that some things seem to be more potentially adaptable than others, and that not everything is possible from every starting point. And the other is, for the traditional vertebrate account, I think we should consider the brain and what it's capable of, not just stop. Well, it's the jaw.
But what's remarkable about the story of the brain is that this really quite complex structure was there so early on, and that it has been able to adapt while keeping that basic structure. As animals, for instance, have moved out of the water and onto land, and then from there into the air, and all these, and then back into the water.
Exactly.
And it's done that without any change in the gross architecture. But there's lots of other changes around that architecture.
But what I'm pointing out is that the neglected part of that original architecture may be something about this sort of multiplication, or having several kinds of, let's call them learning engines, in that structure that may not really have been in a number of the other small organisms at the time. That maybe that's where we might be looking. So the preserved structure in that, in all of those animals, for example, if we take the 4 brain divisions, the thing called the medial pallium is recognizable as the hippocampus in mammals, and is involved in rapid memory and navigation and those sorts of things. And to the extent that there is any large comparative basis, it's the same kind of general function in birds, in the several studies, also in fish. I'm not so sure about what the reptile base is, but here's one part of the forebrain that is remarkably consistent in sort of where it is and what it does over all that range. So I think we can really look at that kind of comparative base in the forebrain now that we really couldn't before.
But you present a very quantitative approach to towards trying to understand these invariant aspects of brain evolution, and you were pointing us to your translating time.net domain, where you present data on 18 different species, and then you use different approaches like regression. You looked at sizes of different brain structures to try to get a handle on, okay, how invariant are these structures and their relations across these 18 species. So what is standing out in that relationship most, in your opinion?
The thing that utterly surprised us right from the beginning, and still does, we still keep making mistakes about hypothesizing the opposite, which is the absolute stability of mammalian brain development. So essentially what this model says and can do is that I can transform the developmental schedule of a mouse, and I'm talking about just the brain here, from the time the first neurons are generated to sort of the start of the very first behavior. And I can simply turn a nonlinear dial and with 99% accuracy predict when that's going to happen in a cat, in a monkey, in a human. And the detail of this translation is very deep. So when I'm saying translate the schedule, I mean I can tell you when the Purkinje cells in the cerebellum are born. I can tell you when the cells in layer 4 of somatosensory cortex are born. I can tell all these very specific things about brain development across all these species with that amount of accuracy. Now this was originally surprising in that I thought that changing, sort of telescoping and compressing or shifting time things would be one of the major ways by which species differences would occur. And we have got a couple cases of that I described, which is in the case of how much cortex you want versus olfactory bulb, and the other was how you change a retina to be nocturnal or diurnal, where you do get shifts of schedules with respect to each other. But overall, that mammalian brain development schedule stays rock steady. You don't change it. This is not my interpretation of these data. That is the data. You know,
so it's nonlinear. It's nonlinear across species or within a species?
So so when I say turn a nonlinear dial, that means in order to transform the very early events of a mouse schedule into a monkey schedule, I don't have to change it very much, but to change, we get onto an exponential curve. But for the late events, like takes first step,
right,
there's going to be much more relative duration between the points in the monkey scale or the human scale than the mouse scale. So there's not so much difference in the first events. There's big temporal differences in the late events, but this is just an exponential predictable curve. Does that make sense?
Right. Oh, so there's just a couple of parameters and you can capture any creature.
Yeah.
But now, do you see this model as providing you the scaffold of a potential brain, or it really defines a brain?
And so I was supposed to describing it in sort of scaffold of a brain. That this maybe is the a stable economical structure that is best, maybe the one that positions itself best to make maximum use of experience. But
that's a positive way of looking at it. Another one might be to say that this is a fixed timetable. You can't mess with it without killing the animal.
I used to think that. I mean, that's a Stephen Jay Gould kind of argument, which is that and I have that title as a contrast in a lot of my papers, which is things like, is it developmental constraint or developmental structure?
Right.
And I keep coming back to 450 million years of defending the same structure.
Right.
Okay. I mean, I think we could have gotten out of it in that amount of time if it was a true constraint.
So we can't do better. I think
peak
in some way.
Yeah. So I'm thinking of this more. I'm trying to push myself into thinking, okay, how do I describe this as an optimal state? What is it optimizing as opposed to what is it? Why are we stuck with it? A lot of these basic body plan things are so actively defended on a genetic level by animals that it looks like not only are they not constraints of the sense of being stuck with a thing, that they're actively kept in place in the genetics of the animal. So that makes it reasonable and also falsifiable too, that these are in some way optimal, and that's a lot of the change in the evo-devo approach to whole body and nervous system stuff is, okay, let's consider that this might be optimal. What is it optimizing?
Right. Yeah.
But but this observation is based on a number of descriptors that you use to look at brain development.
It's all pretty much straightforward gross anatomy.
Sure, exactly.
So,
but then you could make the argument, actually, it blinds you for other kinds of influences that might be more dependent on the environment, that might be more flexible or more dynamically regulated. Or you don't expect...
I really am not making that distinction in any way. So I'm thinking that this is a way that lets you be flexible and dynamically integrated.
Except that you're always on this fixed time schedule.
Yeah.
Right? Okay. There's no way of escaping that one.
Yeah.
Because what I'm sensing is a potential conflict with the more general evo-devo perspective that you also have.
Yeah.
But now we come out with a perspective that says, well, actually, this whole development program is just fixed. There's nothing you can do about it. Environment won't influence that. On the other hand, also Barry Kolb was telling us yesterday about the roles of stress in development. This might be a factor, but maybe you don't see that because the level of description doesn't allow you to extract these features.
I think the talk you presented actually did balance some of that fixed constraint with some flexibility, because you talked then a lot about how delaying timing or delaying the onset of certain things, I guess, but that's within a window. You can't delay things indefinitely, but you can shift things around enough that can have quite big changes.
Yeah. I can, for example, add two things. So one thing I brought up with the olfactory versus visual stuff is a sort of preset for a commonly encountered change in niche in animals that they would have sort of been filtered to have mechanisms to respond to. Stress, you can recharacterize this as we're used to seeing a very stable environment. We're used to seeing a very unstable environment. And so it's interesting to me also that you see a sort of suite of stress responses as part of the, this is below innate. Okay. So this is more in the, the structure of the genome over evolutionary time that has the ability to respond to both kinds of environments. There's another thing I know I tried to get started on, but haven't gotten too far, which is understanding critical periods and plasticity. So, this is in fact one of our current projects. So, I know nothing at this point about what the constraints are on these sorts of things. So, is there really any, say, across mammal optimal period to learn certain kinds of knowledge that are good for particular structures? No one's really looked at it, tried to gather information in that way. Or do you not do that kind of thing? For example, in birdsong, which has been something that's been studied a great deal for critical periods. So, a story for birdsong that's often told is that say the birds in this hemisphere or whatever come north and establish their nest, and the eggs are laid, and the nestlings are in the nest, and they hear song in the spring, and they sort of match up to their template, and they learn the song, and there's a critical period, and the NMDA receptors come on and they come off, and okay, so maybe there's this critical period, but then it turns out that there's an unfortunate set of nestlings who are born in August, and they don't hear any song at all. And so what do? Do you just wasted all that reproductive effort and grew a whole bunch of song? Well, it turns out it isn't like that at all. These animals put their critical period on hold until the next spring, when they actually will hear some song. So this is something where the appropriate kind of input appears to initiate the critical period. So maybe the thing that would be general would not be having a certain time, but a certain kind of initiation. So self-initiated, self-terminated critical periods, as opposed to things that are set in there.
Right.
And so
that would then allow environmental cues to actually trigger a part of a developmental program.
Yeah. And you really have to understand that I'm starting from the position where I thought that everything was in play, and that everything should have been as changeable as that. And there seems to be a whole lot of structural stuff that just isn't.
Right, exactly.
And then we'll see, what happens to things like that.
But now in your description of evolution of isocortex versus olfactory bulb, right, over these 18 mammalian species that you looked at, you saw there's a very specific pattern. At first, all species are closely clustered. It's not that there is some variability within each species clustered, but you also interpret that in terms of some sort of continuum in this relationship between isocortex size, olfactory bulb size, as if it's some sort of trade-off, like more olfactory bulb, less isocortex.
Okay.
Is that really how you see it? A trade-off between these structures?
I showed you two kinds of data. So I showed you data on a whole lot of different mammals that showed a continuum, both across and within taxonomic groups. So that would mean that, say looking within monkeys, we could find some monkeys that have virtually no olfactory bulb and limbic system at all, hardly, and some that have a fair amount. And we can find that pretty much in any one of our groups. Okay. So then we go back to, instead of 180 animals or so, we go back to our 18 that we have the really elaborate developmental data on. And so we can find the examples of the animals that are low and high on our olfactory versus cortex dimensions. And we say, okay, is there a timing component to that? And I can't really talk much about how much they are, or how continuous those animals are. I suppose I could, but I don't think it's really enough data. And so then I can find, if I just ask, let me make a split of these into high and low olfactory versus cortex groups. Is there a difference in how they develop? And yes, there is. So the primates and the carnivores with the high cortex delay producing their cortex until later. And that makes more of it because they have more time to develop their precursor cells.
No, the sticking point for me here is that, is there any kind of intrinsic constraint in this developing brain? That's okay. If I'm allocating more resources to one structure, then let's say there's a metabolic cost, and therefore I cannot grow another structure equally well. So that there's always a sort of, from a pure morphogenesis perspective, there are constraints. On the other hand, you can also say, no, every structure develops as an independent module triggered by environmental conditions, the niche you're in. So in principle, I could grow a huge olfactory bulb and a big cortex if my environment, and actually,
yes, we do have
the carnivores. Some carnivores will do that, right? So where are we in those two interpretations?
So brain is really expensive. So I've actually written a little bit about this. You can contrast two kinds of explanations. So one is, if brain is expensive, then it's reasonable to get the kind of negative correlation that we see there. So if you have a high cortex value, you're relatively more likely to have a low limbic olfactory one. That had, when you have like an energetic or caloric restraint of something like that, would be something you'd find. Another is a mechanistic constraint, which is something I've been looking at. I showed a picture of that showed the fact that the thing that gives rise to the olfactory cortex and the hippocampus are, so the neocortex is sitting right between the hippocampus and the olfactory cortex embryologically, and it looks like it would be just so easy. Let's take that primordial tissue and give it more to the cortex. Let's take that and give it more to the other two, which are immediately adjacent to it. That implies a zero-sum game. That if it's that mechanism and you do it that way, it should always be push-pull like that. Now, the evidence I can offer against that is that it's only mammals that have the negative correlation that I know of so far.
So is that
okay?
Is that particular example? But are you then looking a bit too late in embryology because
there's nothing earlier in embryology than that.
This is really early.
Yeah,
this is because you were talking about the migration of the precursor cells. And this is at that stage.
Yeah, so there is no earlier in which we could actually identify something that's going to give rise to the cortex.
Okay. But one of the really interesting things I think you were showing was the constraint that the developmental process has on the potential for evolution, because you were saying that it was only the lateral parts of this embryological structure that had the potential to really change, and that the more central parts were fairly fixed. Is that right?
Yeah. So this is just a description of the data where if you lay out the embryonic brain on a front to back, middle to edge,
right?
It turns out that how long the cells divide during early embryogenesis depends on position. So the closer you are to the edge, the longer. The closer you are to the front, the longer.
Right.
So this is just describing.
But that description must be capturing some constraint, presumably, that we don't perhaps understand very well.
I don't know if it's a, I don't think it's a property of embryonic tissues to divide a lot at the edge or something. I've never heard of anything like that. But, so maybe there is something like that. But what I would say is sort of a interesting overall interpretation of this is to come at it just opposite. Okay, so what you do is you set up an embryonic structure that gives you variation on some dimension. And in this case, it's variation in the numbers of cells and the duration that structure is going to produce. Then you allocate function to that location. So if you, for example, one thing that's quite interesting about this, there are two places in human brains and mammalian brains that always continue to produce neurons throughout life. What are those? That's the hippocampus and the olfactory bulb. Where are those? Those are sitting on exactly that edge there.
Right.
So if you look at bird brains, they produce neurons in many more places throughout life, but all you move in is just a little bit more towards the midline and pick up the structures that are sitting on that edge. If you're a fish, you're essentially generating all the brain throughout life, but you are generating sort of relatively more of it on those lateral edges. Mm-hmm. So then I think the possibility is that you then take that variation in the size that it's going to be and the potential energetic cost you're going to put into it, and then you can put function into it by designating those cells there in some different way.
Right. But these are precursor cells. They've yet to specialize into particular neuron types. They've yet even to migrate into position. Mm-hmm.
Well, I mean, I show the fate map of this thing is as we're describing it now in vertebrates, very fixed.
Right.
So the medial part is always going to be motor neurons, and the next step over is going to be the visceral motor neurons, and so those locations mean something very specific in terms of what neuron is going to be being generated. Also means a duration. But when this all started out, maybe the fact that assignment of type in the course of development comes after the decision of how long you're going to be generated means that it could be you assigned, sort of type after the sort of size of the thing was entered into the equation.
But I mean, it comes back to this question of how gridlocked is the design of the brain. Mm-hmm. And what essentially we're saying is that there's certain things that are decided early on in development, maybe, and if you tried to change anything there, it might have a lot of knock-on consequences.
Yeah, so there's some of those things, but look, what the things that do get big are the very things that stay out of gridlock. So the ones that are on the lateral edge are pretty much uniformly multisensory, multimotor, can control different effector systems, and are the very parts of the brain, with the exception of the very specific olfactory cortex, that are the most plastic and changeable in their functions. So you allocate more space to the specifically multimodal things, which can be allocated to anything.
Yeah, but there's an interesting consequence of that maybe, because on the one hand, I think it's also important to take into account the morphological constraint imposed by having a skull that you have to fill.
Well, the brain generates the skull, not the other way around. Okay.
But these things develop together.
Yeah.
And there will be also mechanical constraints on that developing brain. And you better lay down your brainstem before you lay down your cortex, because otherwise you cannot pack it in there anymore. So that this already defines a certain logical order, you would think.
Yeah.
But then as you move out later in development, so the more primitive structures are laid down, it's not a surprise you might end up with the most non-specific structures, because these also should be the hyperplastic structure, because they are more dependent on somatic time to wire themselves into that system because they're under constraint, if you want. Right?
Yes.
There's less guidance that you can give them. So maybe this already then tells you why these more cortical-like structures, these hyperplastic, multimodal associative structures, are then more lateral and at the outside of a developing brain, because they're actually, these are easy to specify. And you then leave it to their developmental, to their learning capability to wire themselves up with the rest of the system. Would that make sense to you?
Yeah. There's another way of thinking about that, which is quite parallel, which is what a bunch of computer scientists thinking about control systems have done. And so there's several several groups that have come down on this same kind of organizational principle. So if you want to make a device that can describe sort of catastrophic loss and sudden gains, okay, how do you build it? Well, what you want to do is to keep its basic functions, like getting around and recognizing things, kind of untouched. And the last thing you want to do if you're going to make a big, fancy new brain is to have, your motor neurons on the one hand, they're going to do something, and then you have your sensory neurons, and then between those you interpose some gigantic processing thing. And what you've succeeded in doing is now slowing down this organism so much that it will never, ever survive anything whatsoever. So what people found a much better control architecture to be is to keep those kinds of essential motor organizational functions by themselves, and you build a brain beside that brain.
Right. Okay.
Right. Yeah. And that's what's going on. I think the better description here of so you're building a model of your brain, sort of this predictive thing, and evolution says, oh, that seems like a good idea too. I'm glad you computer-sized thought of that, that you are now being able to plan and simulate and integrate things while still kind of carrying on as usual.
So that means you lay down these midline structures, and then you sort of pad it with the hyperplastic cortex-like structure.
Yeah, and it's literally beside in two ways.
Yeah, exactly.
So it's not, so somehow I find it easier or more pleasant to think about this sort of building a brain beside the brain than just having extra stuff lying around.
Sure.
But perhaps it comes down to the same.
So looking across all these pieces you've analyzed, what would you now see as the blueprint of that mammalian brain?
So we have the whole spinal motor sensory core that takes care of all sort of essential movement and eating and breathing, and then we gonna add on some limbs if we're going to be fancy that are sort of the fundamental operating arrangement. Then I'm very much fond of a guy named Bjorn Merker and his views of how to, but he's getting into consciousness, but we don't have to discuss that so much. But
no, we know Bjorn very well. We just spent 2 weeks with him in Woods Hole. Yeah.
Okay, so great. So he views the midbrain as the place where all this basic integration comes together to make a sort of a sketch of operations for the animal. So what's in front of me? What can I do with it? What do I want to do? Then sort of coming from the other direction, we have the whole visceral brain that knows about, okay, what is my state? What sort of future state would I like? Am I trying to mobilize energy now or save it? Or, what do I want to show other individuals about what my energy state is? And then I'm going to combine that with that same sketch. This is something that's part of the vertebrate makeup and highly plastic. It's going to be quite different from one species to the next, how energy is going to be allocated and how fast and towards what. And then we have these gigantic learning loops sitting around this whole thing. One is the slow auto-associating cortex thing, the fast auto-associating hippocampus, the one who's going to take the output of both those things together, the reinforcement circuitry, and says, okay, which of these combinations actually helped me and which do I wish to repeat as an animal? And then the cerebellar-like circuits that take plans and optimize them. So basically I see this motivational core with these second brains sitting to the side,
right,
and computing the state of that basic operating system, I guess. And that would be it. Yeah.
So 4 second brains. Yes.
Yeah.
Yeah. That's. Oh.
So which animal of all the species that you in your database, which animal then gives us the purest reflection of that blueprint, if you want?
Everyone.
All of them. No, but let's say some might have reduced some parts of it, and they might have exaggerated other parts.
The house cat. I don't know.
Okay.
No, I mean since they, at least the mammals, all are on the same general trajectories. It's really almost impossible to answer that question. I mean,
but one thing that is obviously talked about is the change of size in cortex, perhaps more than changes in these other second brain systems. Do you think too much is made of that?
But it's not the case that it's the so it's on its exactly expected allometric line, as is the cerebellum. A lot of people make a lot of whether we should count neurons or volume or something, or caloric expense. You basically need to count them all. So how many neurons is one measure of how big a structure is. How actually big it is another measure of how big it is, or how many synapses you have, or.
But the Harry Jerrison story was that so-called higher mammals had bigger brains than other mammals.
Yeah. So you can dissociate brain size from body size.
Yeah.
Right.
Yeah.
But you cannot dissociate internal brain structure size from brain size. So a certain brain will always, if it's a primate, let's set the olfactory parameter. Okay. At the outset, we have exactly the size cortex we should have for our size of brain. If we were a dolphin and we have a bigger brain, we have more cortex than us proportionately. So there's no special selection on the cortex.
But then there's the strong claim that in hominid evolution,
it's not true.
Okay.
It's the right size.
So you mean in relative terms, there's invariance.
There was a change in brain size. Overall.
So the thing is that the brain size, we have a really, really big brain for our body size. We have exactly the cortex size that we should have for our brain size.
Okay. But is that a constraint that if you need, just to take the corticocentric view, which I don't actually hold, but, sort of devil's advocate, if I want a bigger cortex and I have these developmental constraints, I just have to build a bigger brain. There's no other way around it.
That's what evolution says so far.
Yeah, so it could be read in that way. If I'm wedded to my view that humans have this fantastic neocortex, and that we just grew extra bits of brain that we maybe don't use so much in order to make that possible.
No, but there's something that there's an inconsistency now here. Because if it's always relative to overall brain size.
Yes. There's a real problem.
Earlier we discussed that you said, no, you can actually have relative differences between a limbic brain, the limbic cortex, and the isocortex.
Yeah, so right? You meant I prefaced this whole thing with, let's set the limbic factor.
Okay. Okay. Right. You were smart. Yes. Good. Okay.
Yes. There's the second component.
Good. Okay. Fair enough. Yes. Okay. Fair enough. Okay.
Yeah.
So that second component says that, my cortex scales with my brain size, but also I can be a cortical oriented species, or I can be an olfactory limbic oriented species.
Yes.
And that accounts for how much of the variance, roughly?
3% of
really? Okay. So
That's 3% is a lot of variance considering the range that we have here.
That's very
a lot of
tiny.
volume. Well, it's a small amount of variance. It's a larger amount of tissue if you're considering the difference between, mediums. Well, I showed a picture of a owl monkey's brain and a goody brain of exactly the same mass.
Yeah.
Mm-hmm.
And so the one is a cortex specializer, and the cortex is hanging all over the side, so you can't see the olfactory bulb and you can't see the cerebellum and all that in the owl monkey because the cortex has overgrown it. But in this very same sized agouti, you get a very good view of the olfactory bulbs and the cerebellum and everything, just because the cortex has,
which is a rodent, right?
Yeah, yes, it's a big South American rodent,
right?
And so 3% sounds little, but if you look at those brains, that's a perfectly impressive difference. Okay.
And I guess, staying with my devil's advocate position, some people would also argue that cortex has become specialized in other ways in primates. For instance, we have 6 layers of cortex like every other mammal, but we seem to have richer networks within some of those cortical layers. I mean, do you buy into any of that? I think you had a slide showing that layer 2-3 was expanded.
But the point of my slide,
richer network and interconnectivity, at least.
But I was trying to show that this is how the gradient of the cortex plays out over different brains. So in the set of animals I have in this set, the human has the largest cortex, but I don't have any dolphins or whales in there, so I can't really say that we somehow, they're primates or anything, hold the prize for most complex network. And I'm not sure exactly what that means, except sort of a self-reifying, we're complex, look at that thing, it's complex.
Well, I think if Henry Kennedy was here, he'll be here next week, and he can contradict this. He would say that primates have this richer, within the layers, they have these circuits that take advantage of these additional cells that you have there. So you have a few sublayers.
I guess so the question is whether this is some virtue of being a primate or virtue of having a large cortex, and that we just don't know yet. Yeah. I mean,
you also said that with respect to, let's say, niche specificity, this might also relate to this point, that actually there are other processes at work as well that might be linked to then how that organism interacts with its niche, which might be control of apoptosis, thalamic drive onto a cortical structure, which might vary, and in general, activity-dependent volume change, right? So that might mean that from a developmental perspective, you have, let's say, a prototypical scaffold that then gets biased by how that niche and the embodiment is sort of driving the scaffold using these 3 principles. Yeah. Would you buy that?
Yeah. I mean, so taking this basic set of layers and then embedding it in different kinds of experience or different kinds of early instruction, I imagine you get all kinds of different things.
But it might then account for these differences. There might not be no contradiction.
Yeah, I think so. And there's a lot of really basic stuff that we don't know. If you look at dolphin cortex, for example, it looks it's sort of it puts a little bit more total volume into area and less into cortex depth. And me and my group and we've done a lot of modeling of this kind of thing, and you have only to change that sort of quit fraction in early development in the cortex, the very slightest amount to sort of direct stuff into more neurons per cortical column versus more area. So I don't think we really know very much yet about just what the consequences of small changes in early development parameters are. And I think, there might, and I've never understood why more layers in a cortex was supposed to be somehow intrinsically better.
More is better.
That's the general argument. But we still have the same hippocampus that everybody else has, and including the fish, and seems to do just fine.
Mm-hmm.
I just don't see. I mean, if someone would come up and show me, and I defy you to find someone who has that, here we have a 5-layered structure and look what it can do, but I'm going to make 6 layers. And oh man, now we got calculus. I mean, I don't think so. So it's really, I mean, it would be nice to actually see some demonstration, but now it's all sort of, large numbers mean something more complex.
Well, you did in your talk describe some changes across cortex, particularly talked about a gradient of increased compression going from the back of the brain to the front of the brain. And then you talked about the front of the brain having more fan-in, more systems talking into the front of the brain. And
which I got from Henry Kennedy's study I wanted to point out.
So, and you can imagine that bigger brains are going to have more steps of compression.
Exactly. You don't have to imagine it, they do.
So by the time you get to frontal cortex, you've got more abstraction.
Exactly.
That's how you get calculus.
Yeah. Yeah, but that you just conflated the...
I'm happy you explained that to us, Tony.
But we were talking about layers. Whether 6 layers of cortex is better than 4 in implementing calculus versus the number of steps that in a sort of hierarchy, which is a different thing altogether. So I can easily make an argument as to why embellishing a hierarchy might be a better thing for extracting more and more abstract explanation. And I wouldn't have to come up with it myself. I could come up with all kinds of computational models that people have made on exactly this point. So that's why finding this hierarchy from, a large number of neurons in the back of the cortex to a very small number in the front, and this progressively greater compression, the bigger the brain gets, sort of maps onto the computational work that people have done really nicely. And so I'm not making fun of the layer thing so much, but there's no comparable literature that says, 5 layers in a column allows me to do something that I couldn't do with 4. I've just never seen anything even take that on.
Right.
So it could be.
But no, in some sense, you also made the point that, if I have sort of this midline controller, and then we have all these added on learning machines, then you said, if you then take cortex, it's sort of equipotential, right?
Starting sort of equipotential, I guess, in the smaller brains.
No, but you also made that point where you said, look, what's special about, let's say, a language area, if you just look at it from, let's say, a morphological, anatomical perspective, is there anything special about it? So you seem to be making this claim that cortex has these sort of, this infinite, not infinite, but this very hyperplastic properties that would allow it to sort of tune to any kind of information that it is exposed to. So equipotentiality is really for you an important principle to understand how the system operates.
Let's sort of go empirical on this. Okay, so if you look across the cortex, the place where you see sort of the most relative diversity in gene expression are also the ones that are not the ancient parts of cortex, but the historically homologous parts of cortex, so that you can see in the same animals all the time. So you can, this is Leah Krubitzer stuff, so you can always find a primary visual cortex. You can always find a primary somatosensory and an auditory cortex in all the mammals. And then if you look at, and so these identifiable regions have had the sort of most time to kind of accrue specific genetic information. Okay. And you see the most diversity in gene expression in them compared to the others. But the thing I think you pointed out, I pulled out in the lecture was, okay, so tell me something in the genetics of primary visual cortex that makes it optimal for being visual cortex other than getting visual information. Now there may well be something, but I have been asking people for a long time now, and not in a hostile manner. I mean, because I really would like to know, okay, if you're going to make this visual cortex, does that mean that it has, the neurotransmitter that's just perfect for and neurotransmitter receptor systems that are just perfect for the normal time course of visual events, or the, axon spread or whatever, something about that would really tailor primary visual cortex to its end? So that's what you'd want to ask. And so far we only have stuff that shows up in the cortex because it gets a certain kind of input. So you see all these structures that almost certainly have some real innate component, but grow as a result of learning and experience. But we don't know if there's anything in vision or somesthesis or audition or something that's specific to those regions, and that makes the analysis of that kind of sensory information better because they typically end up in that particular place.
Or maybe we are also biased in trying to interpret these areas too strongly in unimodal terms.
Yeah,
because you might find also multimodal responses in a visual area,
and you certainly do. And that's the other half of this thing. You see so much stuff just recently about conversion of visual cortex into other uses in reading Braille or echolocation or whatever. And in cases where you don't have to be blind from birth, but can just try to take up a Braille hobby kind of recently. And the one interesting thing is that we're just sort of fixated on visual cortex. I've never seen anyone try to do anything similar. Do people who've lost sensation in their right hand use that for better understanding movies? I don't know. It's just that we don't tend to think of it as a surface that can be invaded so much or something. I don't know.
Right. So then so now we look very much at, if you want, the developmental program. Okay. But you also emphasized very much the role of motivation and embodiment. So how do those factors then really come in to the development and the creation of a brain?
Yeah. So, well, I originally started doing this research because I wanted to find out how you get a brain's wired for adaptive ends. And I wanted to find out, okay, how, if I wanted to be a really visual animal, how would I set that up? And what all this research taught me is that my initial guess about how to do that was entirely wrong. I imagined that you sort of somehow had a way of genetically identifying all the parts of the different parts of the brain and body that were visual, and you could somehow name them genetically and cause them to co-vary, and that's how you would do that. Now I think that you generate this rather determinate in structure but plastic in content brain. And what you do to make a more visual brain is make the animal pay attention to its visual system, particularly as, for example, we like to look at eyes and faces, spend a lot of time learning about that, and then that's what the environmental loop in that, then that is what your brain comes to analyze. So I think if you look at where things really change in the brain from species to species, it's in this sort of basal forebrain, what motivational system is attached to those fundamental reinforcement circuitry. And if you're going to send an organism on a different path, you change what it cares about. and that's what I'm interested in looking at now as I think the central place where sort of the organism and the environment come together to specify what the brain consists of.
And one of the things that I got from your talk was this notion of the adaptable nature of the vertebrate mammalian brain, due to its kind of latent capacity. You know that we've been through this 400 million year history of different species, and that in some way modern brains have retained some of that history, even though you're adapted to some particular environment now. Your ancestors were adapted to very different environments. And you gave the example of monkeys that have adapted to nocturnal living and have, I presume, fairly quickly evolved a lot of nocturnal visual capacities that you might see in other mammals. So am I right in understanding that as being quite a strong claim about the latent capability of the nervous system to, recover these capabilities they've had in the past and roll them out when the opportunity arises?
I wouldn't go quite that far. So I was making two kinds of claims there. One is for things that have been encountered routinely, like nocturnal versus diurnal, or olfactory is of more useful information to me than visual, or the one that just came up as we were talking here, this environment is really stable, this environment is not, the sort of stress dimension. Then in those cases, you may well have the ability sort of retained to rather rapidly and in a coordinated way switch from one mode to another.
Would that be sort of epigenetic in part? I mean,
sometimes epigenetic, and but the ones I were talking about, none of them were epigenetic to my knowledge.
Do you have an example in sort of
the stress
mammals of sort of epigenetic changes? Of that
in the stress kinds of things where it's going to, when a particular kind of early environment is going to send you in a completely different direction for what kind of things you attend to and what motivates you and so forth. But that's not what I do my work on. Then this other, the notion that the rest of the plasticity of the brain somehow embodies every possible thing that an ancestor has done. No, I don't think so.
I don't think
that's a bridge too far. Okay.
But there's a lot of what was used to be called junk DNA. Now people think, well, actually, it's got all this latent potential in it, and that explains some fairly rapid transitions.
It's quite possible. So it wouldn't be the first time. That was that's been. I have had to been so careful talking about anything that vaguely sounds like that with the way biology has been and sort of adaptation. The notion that there could be anything other than the current adaptation state was such an evil thing to say for quite some time that it's taken me a while to get comfortable with even saying something like that out loud, other than in my.
No, because also certainly in popular culture, there's now this Dawkian view on evolution, which just means it's very much sort of feed forward, controlled by sort of these little fragments of DNA that sort of dictate what the phenotype will look like. But now in what you're proposing, it looks like the picture is becoming more complicated. So do you see this really as a drastic departure from this more old-fashioned, the traditional reductionist view, or is it sort of an amendment of it?
I don't really it's the whole deviation that biology took to looking at only genes as the carrier of information at this point strikes me as just strange science. So multi-level selection, for example, somehow that would be to me like saying, well, I can study the properties of molecules, but I can never ever describe the properties of gases, which is just so collectively you can talk about in the kind of statistical language that I'm often using, there's variance that's accounted for at the level of species. There's at the level of taxon. There's all kinds of, it's that the variation, useful variation across animals in anything you want to measure is not attached to the gene. And this is not a question about the usual. I mean, this is one version of multi-level selection arguments. There's one thing is all the causal structure at the level of selection for particular genes. But the second But the second is just a more, generic one. Can I use species to account for example, the relative, predominance of different genes on Earth? Well, I'm sure the snowy owl would say that the presence of human genes on Earth has some sort of consequences for its frequency. And that's the kind of analysis of variance approach to the genome that just doesn't really get thought of. And as people have been talking about how you talk about causation in biology. So, but when people aren't used to considering the different levels of selection too much, and I think people will get better at it.
Okay. So Barbara, you're in the study of evolution and the brain for quite a while, and in some sense, you also have been changing your perspectives on this.
Totally, yeah.
Right. So if we now want to follow in your footsteps, what's Barbara's law that we should adhere to?
There's a particular sort of unsettled or questioning state, which you can either ignore or you can attend to. And that is, there's someone just told me a story, but I'm not really happy about it. And whenever you get that, I'm not really happy about it, pay attention to that, and try to figure out what the causes, of why that story seems inadequate. Really put this major, heavy alert system on in your head for that particular kind of gut feeling, essentially, that something is wrong with an explanation.
Right. So pay attention to annoying surprises.
Yes.
So now 5 years from now, Tony and I are going to come and visit you at Cornell University to check whether you've been able to verify a prediction you're going to make today. So what's the most important prediction that you want to see tested in this time frame of about 5 years?
That particular way of phrasing the question, I need to rephrase a little, because I like to, my prediction is that understanding motivational circuitry and its changes will be the way to understand how species differences emerge. But what we have is a complete absence of information about a lot of that. So people have really just begun. And I really think that for a lot of this kind of for biology in general, we get into hypothesis testing way too soon. And the first thing that you need to do is describe the state of variation that's there. And so I'd be happy if I knew a lot more about what the actual variation was between species in how their motivational systems would be hooked up, and then we'll worry about predicting a little bit later.
Okay, very good. Okay. Barbara Finlay, thank you very much for this conversation.
Okay, thank you.
Thank you. That was great.
Okay.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.